Thank you very much for the invitation, Nevin and Trey, to come to this really interesting meeting. So I'm going to be telling you about the current status of our attempts to get a broad view of the human interactome. This is a project that's been going on for now about 6 years, that's between my lab and Steve Geege's lab, led by a very talented postdoc at Hutland. Before I get into some of the details, I thought I would first, since this is a broad meeting, talk a little bit about what we see as the challenges to decoding interactomes on a global scale. By just pointing out the obvious, there's a large number of human genes. As Emma said, there's on the order of 12,000 different different genes expressed in individual cells. There's hundreds of thousands of proteoforms of different proteins, thousands of interactors, and hundreds of cell types. So how do we possibly come to a definition of the interactome? It's quite a challenge to even think about the complexities of the interactome. But it's even worse than that. And that's because, as you heard from Emma, there is many different kinds of compartments. Each compartment in the cell creates new challenges to try and understand the interactomes that are part of those organelles, those structures. You have to think about different technologies that let you access those individual structures, and not one technique will work for all of them. In addition, everything in the cell is moving. So this is just one very simple example of ER and autophagosomes moving around in the cell, but essentially everything is dynamic. How do we even think about capturing the dynamics of an interactome? And then there's this additional problem, and that is that the proteome varies over about 7 orders of magnitude in a cell. So you have very highly abundant proteins and very low abundance proteins. So you need technologies that let you access both abundant proteins that are perhaps very stably associated, as well as very low abundance proteins. On top of that, you have to think about the fact that there is differences in affinities. You have very stable complexes, you have very transient interactions, and so you need to be able to access the entire range of the proteome, in essence. And so the approach that we'd like to use is interaction proteomics, where in principle you can get down in this low abundance region when you're expressing the protein that happens to be of low abundance. And sometimes you can capture those kinds of interactions quite effectively. So as you know, there are many different ways to map interactions. Early studies used 2 hybrid. There's also correlation profiling, where you look for overlap of proteins in different kinds of fractionation experiments. There's also affinity purification, and then also proximity bitinylation. The proximity bitinylation allows you, in principle, to find things that are close together in the cell but don't necessarily directly interact. It also may allow you to get at dynamic interactions. The methodology we've been using primarily over the last 5 years or so to try and create a map of the human interactome relies on affinity purification. And in our current setup, we use human orpheome as a lentivirus with C-terminal flag HA epitope on it. The initial map we've been working on is 293 cells. We have a system that allows us to create about 500 cell lines a month. And with 3 mass spectrometers, you can just about cover IPs from those 500 cell lines in a month. And then we have a methodology we call Compass Plus to identify high confidence candidate interacting proteins. And so using this system, we've now reported what we call Bioplex 1 and Bioplex 2. These are, Bioplex 2 was the largest experimentally determined interactome available. We now have just about completed Bioplex 3.0. So this is now attempting to IP 10,000 proteins from 293 cells and determine their interactomes. So just a little bit about what kind of data we get. So this is Bioplex 3.0 now, where we have done unsupervised Markov clustering to identify communities within this very large data set of over 118,000 potential interactions. And what you can see is the identification of about 1,500 protein communities. Each little blue dot here is a different community, and the size represents how many proteins are present in the community. And so you can see a lot of, maybe you can't quite read it, but there's a lot of things we know a lot about in this particular map. But what we've been able to do then is use this kind of data to identify new complexes. We can understand the architecture of fitness genes based on CRISPR screens. We can link domains with each other based on different kinds of enrichment strategies. We can predict the function of orphan proteins. About 84% of these communities have enrichments for GO or PFAM terms, so we can understand something about protein function. And we can also predict subcellular localization based on first and second interacting proteins. And then we can also map disease genes on the network. So I'll just show you a couple of examples where we've been able to identify functions of sort of orphan proteins based on this approach. So the first example here is where we took a protein that was previously untouched, sort of the dark proteome. It's called GAT-SL3. To make a long story short, we identified components of the gator complex coming down with GAT-SL3. Again, this protein had never had any data on it in the literature at all. And ultimately, we were able to show that this is actually the arginine sensor that regulates TOR on the lysosome together with David Sabatini's lab. And so we renamed the protein Castor, which is the cellular arginine sensor. In a very similar kind of fortuitous experiment, by IPing various components of the Gator and Kickstar complex, as well as this previously unstudied C7orf60, we were able to, again with the Sabatini lab, identify what turns out to be the S-adenosylmethionine receptor, again regulating mTOR activity on the lysosome. So this is just two examples out of many where there's completely unstudied protein, and we can identify potential interactors and then lead to an understanding of function. Okay, so the question I want to now address for the rest of the talk is, can we begin to explore the diversity of interactomes across cell lineages? So we wanted to be able to get a second interactome in a different cell type. And so we spent a fair amount of time testing different cells, determining their total proteomes, trying to understand what the diversity of proteomes were for cells that we thought we could potentially use in the lab to get a second network. And so I think it's sort of obvious here that you can have situations where in one cell line there might be a very abundant protein that's basically not present in some of the other cells. And so you might get an interactome for any particular protein that's present in those cells, but you may not see it in other cells, right? So there's just cell line specificity in terms of what kind of interactions they might have. To make a long story short, after doing a variety of experiments, we decided to pick HCT116 cells, which is a near diploid cell line, unlike the 293s, which are highly aneuploid. And we wanted to use that cell line to try and build a second network. So I'm just giving you an update now on where we are with the second network. So we've now completed 5,500 IPMS experiments for HCT116 cells. So the network emerging from this is now bigger than Bioplex 2 was. And so you can see here the number of baits relative to what we did with Bioplex 3 in 293 cells. It turns out that there are a lot of shared interactions. Of course, there's not every interaction is shared. There's a lot of cell type specificity here with expression. Also, there's a difference in the levels of expression. We tend to get lower levels of expression with HCT, which might change the ability to detect certain kinds of interactions. But suffice it to say that we've built a network now with 71,000 interactions in HCT cells and are now sort of trying to understand what this network can tell us. So we think that this overlapping set of proteins represent core interactions that are conserved between different cell lineages. Just a few bits of information here. So there's about 3,600 shared baits that lead to this overlap, about 36,000 interactions and 8,000 proteins. And so this is what we call the replicated bioplex, and this is already a pretty large network. And so this is, to my knowledge, the largest comprehensive experimentally replicated network. What does it look like in terms of quality? So I'm showing you now the 293 cells and HCT cell networks looking at quorum complexes. And so we get very high degree of coverage of many of the core complexes in the cell, which is good. It's just sort of stating the obvious that we can get these sort of well-assembled, well-behaved complexes quite easily. And if you look across the entire quorum collection, you can see we do pretty well in terms of identifying the fraction of shared edges in the two different cell lines. Now, quite interestingly, it turns out that there's basically about 500 or so uncharacterized genes that are present in the replicated network. And so previously, whenever we had an unstudied gene and we got something down, we usually couldn't tell what was interesting about it because we didn't have any information with which to judge whether or not it was real. But now we have quite a large number of complexes where we've essentially replicated it in a different cell line. Okay, so one of the main things that we want to be able to do by looking at other cells is to understand how different post-translational modifications may affect interactions, how proteins may change their localization, what can we learn in terms of the dynamics. And then also, as you know, there's many different kinds of stimulus that will change the activities of proteins. And so at a first approximation, some of the unique interactions that are present in either the 293 cells or the HCT116 cells might represent something to do with the sort of dynamic proteome or things that are different in different kinds of cells. And so just a couple of examples of the things that we're sort of finding. So we can find examples where we detect an interactor in HCT116 cells, but not in 293 cells, and there's several different examples shown here. We could also have examples where we basically detect no interaction in the 293 cells or very few interactions, but detect a network in the HCT116 cells. So this is now basically a work in progress. We're trying to understand the HCT116 community in more detail. We just recently got all the data. So I'm just showing you like a couple of different examples. And this is the last example I'm going to show you where we are looking at a protein called TRIM28, which turns out to be a transcriptional regulator that binds to Crab domain proteins. And so what we found is that there's a large number of Crab domain proteins that bind in 293 cells. There's also a large number that bind in HCT116 cells, but we didn't detect those in 293 cells, and then a set that are found in both. And so it turns out that this relates to the protein expression in the cell. So the HCT116 cells have a much more diverse set of Crab domain proteins that are expressed, and we can actually detect those. So Crab domain proteins are sort of interesting in that they're setting up different transcriptional complexes to regulate different kinds of genes. And so what this is sort of suggesting is that you have these two different lineages that are using different sets of Crab domain proteins together with TRIM28 to control expression of the proteome. And so I think that there's going to be a very large number of other examples that emerge as we continue to compare the proteomes of HCT and 116 and continue to build the network. So in summary, we've been working now for about 6 years to establish this robust pipeline. It works pretty well in the cell lines that we're using it for. We have thousands of novel interactors that need to be prosecuted and studied in some way. There is, we think that the overlap between the HCT116 and the 293 cells is going to be a very sort of high confidence, experimentally validated set of data that people can go in and mine. But then we sort of need to think about, well, what can we do in the future? And so I listed here sort of what we're trying to do now. So we want to complete the HCT116 network. We want to be able to expand cell line coverage. We want to be able to expand the clones beyond the 10,000 clones that we have available currently. And then one thing that I didn't mention, there's a huge sort of gap in our understanding of membrane proteins because of the difficulty in doing IPMS the conventional way. And so using other approaches such as proximity biotinylation may provide insight there. And then we're also trying to understand how phosphorylation is controlling things. We've made the data available through multiple different platforms. As you can see here, we have our own Bioplex website. The data goes into BioGrid, oftentimes pre-publication, and then Trey's network also has it in it. And so I'll just stop here and just mention all the people whose names are in red are the current people working on the project. And this has been a close collaboration with Steve Gigi's lab. So thank you very much.
Thank you, Wade. Do we have any questions?
Great talk, Wade.
Great.
With respect to the cell type specific interactions, I know this may be hard to give a concrete number, but how much of the differences do you think are owed to expression differences?
Yeah, that's a hard number to actually come up with. I think what we need to do is, let's say we identify a set of 100 interactions. We need to go back in and really test those. Because the problem with mass spectrometry is because it's stochastic, right? You can have an interaction that you can detect in 293 cells, maybe because the bait level is 2 times or 3 times higher than what you have in the HCT, but you see the interaction in the 293 cells, but you just don't see it in HCT. So is it a real negative, or is it that you just didn't detect it? So I think there's a huge bottleneck there in trying to understand how different the interactomes really, really are. So going back and trying to study them one by one will be probably required, but also very slow to actually deduce how specific things are. We can get an idea based on the protein abundance as to whether, we just can't detect it, or if it's not there, we're not going to detect it. But we need to do a lot more analysis.
I have a question in between there related to that. So the proteins that you do not detect in one of them, so do you see any general trends? Is there a certain type of proteins or related to some biological, anything other than abundance?
Well, we probably haven't had enough time with all the data to really know for sure. My guess is it's going to be a combination of abundance and detectability. It might vary from complex to complex or protein to protein as well. Also, this idea of things being in membranes, those might just create a much harder set of targets to go after.
Wade, nice talk. So one of the things that I didn't notice on your last list is interrogating mutants in a way.
So, yeah.
So how can you incorporate that? Because this seems like it's set up beautifully for.
Yeah. So the way that we have been doing that is by using multiplexing mass spectrometry approaches, where we express wild type and mutant proteins, and then we can do IPs in a quantitative manner. So there's many different variations that are interesting to think about. One is, what do the interactomes look like of your disease proteins? And how do they change? And then the other thing is, how do interactomes and dynamics change globally when you have a particular point mutant in your cells, that you're not necessarily interrogating the point mutant directly, you're interrogating what happens to the cell. And so I think you can do different kinds of total proteome analysis, et cetera. We're not going to be able to do interactomes at that level, but you could do focused experiments.
Okay, thanks, Wade, for a great presentation.